The overall goal of this procedure is to study mechanisms of leukocyte recruitment across human liver endothelial cells in the presence of physiological levels of shear stress. This is accomplished by first preparing a confluent monolayer of human hepatic sinusoidal endothelial cells in a microslide, which is subsequently stimulated with inflammatory cytokines. The second step is to isolate lymphocytes from peripheral blood. Next, the microslide is connected to a flow assay system and perfused with lymphocytes in the presence of physiologically relevant shear stress. The final step is to visualize lymphocyte interactions under conditions of flow with a phase contrast microscope. Ultimately, the offline analyses of phase contrast images acquired during the flow assay are used to compare the effect of various inhibitors on lymphocyte recruitment across liver endothelial cells.
This method can help answer key questions in the hepatology field, such as which adhesion protein and chemokine receptor combinations are important for the recruitment of diverse immune cell populations across the sinusoidal endothelium and into the hepatic parenchyma.
The implications of this technique extend to the therapy of chronic inflammatory liver disease, the majority of which are driven by lymphocyte recruitment into liver tissue. Understanding the molecular mechanisms could lead to new anti-inflammatory therapies.
Though this method can provide insight into the recruitment of inflammatory cells to the human liver, it can also be applied to other cell types, such as endothelial cells isolated from an umbilical vein or other vascular beds.